My name is Adriana Sanchez, and today I'm going to talk about a little bit of a project we have in conjunction between Universidad del Rosario and Wake Forest University in the US. In this project, we're trying to understand the potential effects of climate change on the physiology of plants. In this particular case, we're working in the Alpine of Colombia, in the páramo, in Chingaza National Park, and we work with two plant species that are very common in this páramo. And these plant species are chusque and Aispeletia. We record several climatic variables such as temperature, humidity, light, precipitation and cloudiness, as well as several physiological variables such as carbon fixation or assimilation, stress by temperature, radiation, and stress by water. We also put sensors in the plant leaf that tells us how much temperature and how much light are incident on these leaves. As part of this project, as I said before, we're trying to understand the potential effect of climate change. So with the data we have right now, with the climatic variables and with the physiology of these plants, we can project what would happen in time if the scenario of increased temperatures, increased precipitation, or increased cloudiness could happen. So therefore, we could have an idea of what would be the effect on the plant physiology. First, I'm going to explain how we record the light conditions, the radiation. So we use a data logger that records data every 5 seconds. And attached to this data logger, we have a quantum sensor that records all the visible light. And we have a pyranometer that measures all light, like all the light that comes to Earth. With these measurements, we have an idea of how variable is the light here in the páramo. For recording temperature and humidity, we're using this technology that is called iButtons. The iButtons log data every minute and they store it in an inside memory that then you connect to the computer and download all data. To record leaf temperature and light at the leaf level, we're using this technology by Campbell Scientific. We're using a multiplexer for the temperature sensors, and we're using a data logger to which the temperature sensors and the light sensors are connected. Since temperature doesn't change very fast, we record temperature every minute. Light conditions, though, are very variable in the páramo. Therefore, we measure light at the leaf level every 5 seconds. The temperature sensors I mentioned before are connected on the lower side of the leaf, or the abaxial side of the leaf, and we call them thermocouples. The light sensors are connected on the frontal side of the leaf, or the adaxial side of the leaf. They record data every 5 seconds, while the temperature sensors every minute. Now I'm going to explain how we record stress by temperature or by radiation. For that, we use a fluorometer. The fluorometer sends a pulse of light to the leaves and then records transmittance. And the amount of light that the leaf can capture, specifically the photosystem, the second photosystem, gives us an idea of how much light the plant can process. To record the fluorescence, we use this clip. We connect it to the leaf. Then we put the fiber optic cable. We send a pulse of light that you'll see here, and then the machine throws several data. The data we're interested in is this ratio, the FBFM, that in this particular case is 0.82, which means that the plant can use about 82% of the light we send. That means that this leaf is not stressed by temperature or radiation. For recording how much carbon the plant is fixing, we use this machine. It's called a Lycor 6400. It's a very complete system. It's a closed system, and we know how much CO2 we input in the plant. So in order to use it, we use a transparent chamber. We put the leaf in, and then we can record how much light is in the environment, how much light the leaf is experiencing. We know the leaf temperature, and we also know how much conductance the plant is performing. With the 6400, we can measure several things. As I mentioned earlier, it's a closed system where we input about 400 units of carbon. The sample, the leaf, is taking about 3, is the difference between these two. That translates in a photosynthesis of about 1.7. We have a conductance value that this is right now is not correct, it's too high because the leaves are wet, it's been raining too much. But we can tell, for example, how much light the leaf is experiencing. Right now it's about 40, while the environmental light is about 100. Now I'm going to explain how we measure water stress or the water potential of the plants. First, we have to collect a leaf. In this case, this is a leaf of a sapling of Espeletia or frailejón. We put this leaf inside a collar. We tighten the collar around the petiole. So we cut this leaf at the base of the petiole and we put it inside the chamber. This chamber is connected to a nitrogen tank that is going to provide pressure, gas pressure. So this is a direct relationship in which if you put more gas, if you apply more pressure, the plant is going to be more stressed. So we open the rate, we provide gas to the system. And as soon as we see a little water coming out of the petiole, we record the data. In this case, it's 0.5 is the measure of the pressure we're applying. This means that close to zero means that the plant is not stressed at all because it's been raining a lot. The project we have here in Chingasa National Park is going to be compared with a similar project we have in the US. In the US, we work in the Rocky Mountains and the Appalachian Mountains in conjunction with High Point University. The idea is to compare alpine plant life in different parts of the world, right now in North America and South America. With the climatic variables and physiological variables, we can project how climatic change will affect plants in different parts of the world and have a more complete idea of these potential effects.